Welcome to all students. Myself, Om Prakash Godara, Assistant Professor, Pro Physiology, Department Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth, Women University, Jaipur. Today we discuss about our topic is fat metabolism, fatty acid breakdown and synthesis. Mainly जो fat है उसका जो metabolism है वो किस तरीके से होता है और जो fatty acid है उसका सिंथेसिस एंड ब्रेकडाउन किस तरीके से होता है तो देयर में भी डिफरेंट मेथड है इसके अंदर मेनली फॉर्मेशन ऑफ ग्लिसरॉल एंड प्लांट के अंदर जो ग्लिसरॉल है उसका फॉर्मेशन होता है बट वन ऑफ द वेरी कॉमन मेथड इज फ्रॉम डाइहाइड्रोक्सेस्टोन फॉस्फेट व्हिच इज इंटरमीडिएट ऑफ ग्लाइकोलाइसिस पहले जो ग्लाइकोलाइसिस है उसके अंदर डाइहाइड्रोक्सेस्टोन फॉस्फेट है उसका जो फॉर्मेशन होता है तो इसके अंदर इन्वॉल्व होता है फर्स्ट रिड्यूस टू अल्फा ग्लिसरोफॉस्फेट ये मेनली इसका जो फर्स्टली रिडक्शन है वो अल्फा ग्लिसरोफॉस्फेट के अंदर होता है बाय एंजाइम ग्लिसरॉल 3 phosphate dehydrogenase coenzymes NADH2 is oxidized in this reactions mainly coenzyme NADH2 है उसका जो ऑक्सीडेशन है वो उस रिएक्शन के अंदर होता है और अल्फा ग्लिसरोफॉस्फेट इज द हाइड्रोलाइज्ड अल्फा जो ग्लिसरोफॉस्फेट है उसका हाइड्रोलाइसिस होता है मतलब H2 के द्वारा ये हाइड्रोलाइज होता है बाय ग्लिसरॉल फॉस्फेट to liberate phosphoric acid and form glycerol. तो इसका मेनली जो रिएक्शन है उसका ग्लिसरॉल का प्रोडक्शन होता है synthesis of fatty acids long chain saturated fatty acids are synthesized in plants from active 2 carbons units the acetyl coenzymes CH3CO although the reactions of beta oxidations of fatty acids are reversible. The fatty acids are not formed simply by the reverse reactions of beta oxidation. After that, mainly beta oxidations, synthesis of fatty acids from ester coenzymes take place step by step. In each step, the fatty acids chain is increased by 2 carbon atoms. Each step involves 2 reactions. In the first reactions, which take place in presence of acetyl coenzymes carboxylase, acetyl coenzymes combine with CO2 to form melanin coenzymes. Malonic acid is 3 carbon compounds. ATP provides energy while manganese ions and biotins are required as cofactor. Melanin coenzymes react with another molecules of acetyl coenzymes in the presence of fatty acids synthetase and coenzymes NADPS2 to form coenzymes derivatives butyric and acid contains 4 carbon atoms. 1 mole of CO2 S2 and coenzymes A are released while NADPS2 oxidized in the reaction. Butyryl coenzymes in the next step will combine with the melanin coenzymes to form coenzymes derivatives of fatty acids containing 6 carbon atoms. This process is a repeat till coenzymes derivatives of long chain fatty acid which may contains up to 16 to 18 carbon atoms is produced. As a matter of fact, the enzymes fatty acid synthetase is not simple but a complex of many enzymes, multi-enzymes complex, and an acyl carrier proteins called SCP. And actually, the reactions described above only summaries a number of reactions involved in synthesis of fatty acids from acetyl coenzymes, which can be grouped under 3 categories. Firstly, initiation reaction in these reactions, acetyl coenzymes transfer in its acetyl groups to one of the sulfothio groups of multi-enzymes complex fatty acid synthesis. Unsaturated fatty acids are synthesized by denaturation of saturated fatty acids. ACL carrier protein is similar to coenzymes in having phosphopentathions as the functional unit in their structures. In coenzymes, it is a chain consisting of 81 amino acids and esterify adenosine 3,5 bisphosphate, but in ACP it is esterified to serine of proteins chain consisting 81 amino acids. Chain elongation reactions in which 6 different reactions involve here melanin transfer, condensation, reductions, dehydrations, reductions and acyl transfer. Chain elongation start with the transfer of melanin group from melanin coenzymes to second sulfothio group. of the multi-enzyme complex. Then there is condensation of the latter so that C4 carbon unit is produced. This unit by next three reactions, reductions, dehydrations, and reduction is converted into saturated 4 carbon unit. Example, butyryl conjunction. In acyl transfer reactions, the fatty acid reduced is transferred back to the sulfonyl group to which acetyl group was transferred into initiations reactions. The cycle is repeated again and again with melanin transfer, condensation, etc. till the fatty acids residue consists of up to 16 to 18 carbon atoms. Each such turn elongate fatty acid chain by two carbon atoms. Details of chains elongation reactions are termination reactions. When the fatty acid residue has attained a desired length, the chain elongation stop at reactions and cycle is not repeated. The SL group instance of being transferred to the sulfonyl groups of the enzymes is transferred to sulfonyl group of coenzymes molecules. Thus, coenzymes, a derivative of the fatty acid, is produced, which can then be utilized in the fat synthesis. The enzymes become free. After that, it is believed that during this process of fatty acid synthesis, the SL group of fatty acids bound to the sulfo SH group of SL carrier proteins. The latter then passes is from one enzyme of the complex to other. After that, condensation of fatty acids and glycerol. The fat or glycerols mainly it is converted into different type of chemicals and fat-free fatty acids, but from alpha glycophosphate and coenzymes A derivatives of fatty acids. Fatty acid coenzymes are reduced first. There is acylation of alpha glycophosphate by two fatty acyl coenzymes A molecule to form phosphatidic acids. Now, dephosphorylations occur in the presence of phosphate and triglycerides is formed. Acylations of free OH group of glycerides complete the biosynthesis of triglycerides or fat. Fatty acid synthesis and breakdown. Fat are heterogeneous group of molecules. They contain atom of carbon, oxygens, and hydrogens. Fat and fat-like molecules are generally called as lipids. They are insoluble in waters and soluble in organic solvents. Fat serve as a reserve food materials primarily in seeds, whereas the fat-like materials, mainly phospholipid and glycolipids, are constituents of all cell membrane. The cuticular wax are also lipid but are quite different in their compositions from the fats. The complete metabolism of fat leads to oxidations to CO2 and water and the liberations of energy equivalent to 9 calories per gram of 1 gram fat. Biosynthesis of lipid in which the basic lipid unit is a phosphatidic acid which is synthesized from glycerol and fatty acids. Glycerol is synthesized from glyceraldehyde 3-phosphates from glucose. The fatty acids are synthesized from acetyl coenzymes. Fatty acids are also the precursor of fats. Phosphoric acid may form glycerides, neutral fats, or phospholipids by interacting with choline or glycolipids by interacting with sugars. The number of chloroplast and mitochondria contain complex lipid molecules in which fatty acid consists of even number of carbon atoms. Fatty acids may be saturated and unsaturated fatty acids. Example, palmitic acids and stearic acids. Palmitic acid is commonly found in vegetable fats, palm oil, but linoleic acid is in linseed oils present. Anabolism of saturated fatty acids. Fatty acid synthesis is a characteristic of all living organisms. Multi-enzyme complex referred to as a type first fatty acid synthesis are essential for fatty acid synthesis. The first reactions of the sequence is carboxylations of acetyl coenzyme A to malonyl coenzyme A in the presence of enzyme acetyl coenzyme A carboxylase. one molecule of acetyl coenzymes A and one molecule of malonyl coenzymes A are converted to their corresponding acyl proteins derivatives in the presence of the enzymes transacyclases. Anabolism of unsaturated fatty acids. The fatty acid synthesis generally is a palmitate, which is 16 carbons compounds fatty acids. Unsaturated of fatty acids occur in both the mitochondria and endoplasmic reticulum in presence of enzyme fatty acyl coenzyme desaturated. Anabolism of triglycerols. The most important route of triglycerols biosynthesis is Kennedy pathway is as illustrated mainly breakdowns of fatty acids, catabolism of lipids, the first step in plants where odd number of carbon-containing fatty acids are present. By this process, the long chain of fatty acid is gradually broken down until it reduced to 12 carbon atoms. Fatty acid with less than 13 carbons are not affected by this process. One complete alpha oxidation result in the elimination of one carbon atom in the form of CO2 from the CWH group of fatty acids, which alpha carbon atoms number 2, which is adjacent to CWH, is oxidized. Alpha oxidation take place as follows. The fatty acid is oxidatively decarboxylated in the presence of fatty acid. Omega oxidations it occur in the endoplasmic reticulum rather than the mitochondria, the site of beta oxidations. The omega carbon in fatty acid is the carbon fastest in the alkyl chains from the carboxylic acid. In the omega oxidation pathway, this carbon is progressively oxidized first to an alcohol and then to a carboxylic acid, creating a molecules with a carboxylic acid on both ends. Glyoxylate cycles in plants, the glyoxylate cycles occur in special peroxisomes, which are called glyoxysomes. This cycle allows seed to use lipids as a source of energy to form the shoot during germinations. A lipid store of germinating seeds are used for the formations of carbohydrates, the fuel, the growth and development of the organism. The two enzymes which regulate these cycles are isocyclase and malate synthetase. Active breakdowns or degradation of fat take place mainly in this process. During the terminations of fatty acids and decompositions, product may enter into glycolysis and Krebs cycles to release energy and also synthesize soluble sucrose through glyoxylic acid cycle. Then translocated to the growing regions of the germinating seedling. In plants, when carbohydrates reserve declines, fat may form the respiratory substrates to release energy through oxidation. Fat are first hydrolyzed in the presence of the enzymes lipase to yield fatty acids and glycerols. Oxidations of glycerol react with ATP under the influence of glycerol kinase to form glycerol 3-phosphate, which is then oxidized to produce dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase and NAD. Glycolate, glycolytic pathway, this conversion of glycerol to pyruvic acid take place in cytoplasm, which yield to ATP and to NADH, which in turn produce 4 molecules of ATP. If pyruvic acids enter into Krebs cycles, it will produce another 15 ATP moles. Thus, a total formations of ATP is equal to 21, with the consumption of 1 molecule of ATP in the presence of glycerol kinase enzymes. Therefore, net gain of 20 ATP per glycerols. So we discuss about fat metabolism, fatty acid synthesis and breakdowns. तो फैट है mainly क्या होता है कि जो प्लांट है इसके अंदर जो जर्मिनेटिंग सीड्स होते हैं उसके अंदर जो फैट है वो एक एनर्जी मेटाबॉलिज्म के लिए रिक्वायरमेंट्स होता है तो इस कंडीशन में mainly जो फोटोसिंथेसिस जो जर्मिनेटिंग सीड्स है उसके अंदर mainly जो क्लोरोफिल कंटेंट है उसका फॉर्मेशन नहीं होता तो इस कंडीशन में mainly जो फैट है तो उसका जो मेटाबॉलिज्म है वो मेनली फैटी एसिड सिंथेसिस फैट ब्रेकडाउन और जो ग्लाइकोलाइट्स पाथवे मेनली कंप्लीट होता है, और इसके द्वारा मेनली जो एस्टाइल कोएंजाइम्स मॉलिक्यूल्स है उसका फॉर्मेशन होता है। एस्टाइल कोएंजाइम्स मॉलिक्यूल्स क्रेब साइकिल में एंट्री करता है और तो फिर उसका कंप्लीट ऑक्सीडेशन होता है और एटीपी का फॉर्मेशन होता है। इस प्रकार तो मेनली जर्मिनेटिंग सीड्स है उनके अंदर जो एनर्जी है वो मेनली फैटी एसिड सिंथेसिस के द्वारा और इसके जो एंट्री फ्रैक्चर होते हैं इनको एनर्जी मिलती है, तो वो ग्रोइंग पार्ट तक जो एनर्जी है मूव करती है और इनकी ग्रोथ इंक्रीज होती है। थैंक यू स्टूडेंट्स, योर टॉपिक इज